This is a production of Cornell University.
Thank you so much, Don, for such a kind introduction. And thank you so much, everyone, for coming here today. I know it's such a high demanding labor in the field right now, but thanks to the rain that it can gather people to here. So the main goal of my research is only one thing. It was trying to breed switchgrass to be resistant to diseases caused by Bipolaris oryzae. So the topic here that I put is pretty simple, that the switchgrass versus the Bipolaris oryzae, which is a fungal pathogen, a challenge in breeding for resistance. I know that I have been presenting you about switchgrass for so many years and so many times now, but you may think that switchgrass is still out of your circle. It's just out of your everyday life, but actually it's not, because the first picture, this is a switchgrass that I was taking. Was taking this picture right in front of Cornell Health Center or Gannett, and it was planted in the slope, like right in front of that. So switchgrass has been used for preventing the soil erosion pretty well in this case. And also the second pictures, can you guess where is this one? It's in Stewart Park, one of your favorite place in Ithaca. Actually, we have, yeah. So it was planted in just the side of the road. So switchgrass has been used for the landscape decoration quite some time, but actually switchgrass has been introduced as a forage purpose crop. But in 2000, switchgrass has been introduced as one of the biomass candidate by Department of Energy as a biomass crop because of its high yield, and it's also good in terms of greenhouse gas emission capacity right here. It's also very well, it's a native grass to North America, and it's very well adapted across the latitude. So it's separate into upland switchgrass and lowland switchgrass. So the next picture is a comparison between the upland and lowland switchgrass. So on your left-hand side is a upland, and on your right-hand side is a lowland. So normally upland is well adapted to the cold winter, like right around here. But the lowland is well adapted into like the higher moisture area and also provided more yield. Another difference is the ploidy level are different. That upland is normally octoploid and lowland is normally tetraploid. But if you just want like just a general idea, if you walked in the field, so you can separate it by just the colors of it, because the lowland type is normally bluish green. And based on this difference, it's also a difference in terms of the resistance also. Surprisingly, after domestication in 1970s, there are only about 1,700 publications of switchgrass, and little of them, just only 28 of them, is related to disease. Even though John mentioned that there's a lots of diseases in switchgrass, such as rust and anthracnose, and of course, the disease by Bipolaris oryzae. And let's get back to our basic pathology triangle, that to confer the resistance or susceptibility, the host, which is switchgrass that I already introduced to you, was attacked by a pathogen, and in this case is Bipolaris oryzae. Bipolaris oryzae is an ascomyces fungus that it's a necrotropic pathogen. And to present in general, if we want to emphasize the importance of the disease, we always put the worst case scenario. And of course, switchgrass, and of course, Bipolaris oryzae also has the worst case of this disease, that it killed 2 to 3 million people in India during the Bengal famine in 1943. And because this can cause the disease in rice. And this pathogen also causes disease in switchgrass also. It can cause 2 diseases. First is Bipolaris seed rot. As the name suggested, it reduces the germination of the seeds. But even though that seeds can germinate, this pathogen can infect the shoot and the root of the seeds also. And these are the pictures of the hyphae and the cornidia, which is a sexual spore of this pathogen on the seed surface. Another disease is bipyrate leaf spot, that it causes just the necrotic lesions on the leaves like this that can reduce the yield up to 50%. And that's a huge amount if we consider to use switchgrass for biomass production. In terms of management of the disease, we need to understand the life cycle of the pathogen before we make any decision which way we're gonna manage the disease. And Bipolaris oryzae is the seed and soil-borne pathogen, that the first inoculum of the disease is from the seeds, that it was introduced to the field, and after that, the hyphae of this pathogen can grow into the soil and leaf and overwintering within the crop residue in the field. And after that, it can spread, spread the cornidia spore into the mature plant and cause the leaf lesion. The last angle of this triangle is environment. What is the best environment for this pathogen to cause the disease in switchgrass? And of course, it's here in New York with the prolonged high humidity and high temperature during summer. This is the perfect home for this disease. That's why I work with this disease, and why I pick the breeding as a management method over other approaches. That, okay, if it's a fungal disease, why don't we just apply the fungicide to the field or fumigate the soil as it's the soil-borne pathogen? Of course, you can do that, but it's pricey, right? Because we consider consider using switchgrass for biomass production, and we try to reduce the input as much as we can. Another solution is, because it's a seed-borne pathogen, why don't we just soak the seed with the fungicide? Even though I show you that there's a this fungus can grow on the seed surface, but this also has been proved that can be inside the seed tissue. So this may not be an option. Another one is just the cultivation practice. Normally, we can plow the soil to disrupt the life cycle of the disease, but because switchgrass is a perennial crop, so this is not an option. Another concern about this switchgrass is the target environment for switchgrass. It's in marginal land, such as the reclaimed mine area, that we try to reduce the competition between food and fuel in crop to grow in a good land. So this is the target environment here, that it has low nutrient, high heavy metal, and high acidity. That's the worst place to plant switchgrass, but that's the place that we picked for planting them. So, and that's why I picked the method of breeding to manage this disease. So the content today, I already introduced you the background of the host switchgrass, the pathogen of the Bipolaris oryzae. And to make the selection, I set up the screening method first. Let's move into the first one. So for Bipolaris seed rot, I basically just soak the seed with the inoculum and compare with the seed with just the control one. So I set up the screening method based on the variation, and I found that at week these are the and I found that the first week shows the highest germination, but that is not the day that I should collect the data, because do you remember that these pathogen can infect the shoot and the root of the early germinated seeds. So that can drop the germination during the week 3 here. So I picked the week 4 to evaluate this disease and to screen the resistant with this condition. Another disease is the bipyrate leaf spot. So I have 2 options here, to screen in seedlings or to screen in mature plants. I picked the seedlings one because I can handle it in greenhouse any time of the year, and I also can increase the number or increase the replicates as many as I want. So I use at least 1,000 or 2,000 per cycle for screening. That it will take a lot of land to produce in the field for mature plants. So the basic method for screening this is I screen the resistant to bipyrite leaf spot in 4 weeks old seedlings. I sprayed it, cover with a clear plastic bag for 24 hours, release it, and I set up, and I determine the condition when I'm gonna evaluate the disease and what concentration I use. So these are the score that I use to evaluate the disease resistance from 0% to 100% as like 100, the cover the whole leaf. So I have two options to just evaluate at only 1 day or as a percent leaf coverage by the lesions, or I can use the area under disease progress curve along with from day 1 to day 10. And I found that there's a very high correlation between just the 1 day at day 7 to that 10 days value at around 0.9. So the screening method will be just evaluate at the day 7 only. And so I use this criteria and I use the highest concentration to screen this to increase the intensity that you have to survive from this and you'll be the best. And at this point, I was wondering like, if the seeds that was inoculated before that can survive from those pathogens can also increase the resistance when they grow up as seedlings. So, but based on that experiment, I found that there's no correlation between the resistance of these two diseases. So based on this no correlation, I use this no correlation to set up, I use this information to set up the breeding scheme. And let's moving to the third one that I make the actual selection based on these resistance. So basically I use a recurrent phenotypic selection for 2 cycles of selection. And based on the fact that these 2 resistance has no correlation, I separated into resistant bipyrite seed rot and bipyrite leaf spot. I made for 2 cycles of selection. And at the end of cycle 2, I taste the reciprocal taste between that. The one that selected for bipyrite seed rot also got sprayed by the got spray when it was in seedlings also. And because shelter and caving rock show pretty well adapted to New York and high yield in New York State, I make the selection based on these 2 populations, which is Upland, Switchgrass. I produce the half-tip seeds in greenhouse and in field nursery. I also produce so I made a selection for the caving rock and shelter, right, which is an upland type, so I can produce it in the field easily. But I also produce the half-tip seeds of the canlo in the greenhouse also, because canlo is so I use canlo as a lowland representative in this case to have an idea like what is the resistance in the lowland should be. And normally when we make a selection, it's good to know how heritable the trait is. That's why I evaluate the heritability based on the individual and based on the half-tip. And the results showing that for the resistant to bipyrite seed rot, the callow switchgrass showing like pretty high, like moderate to high heritability at 0.5. And the one that I make that actual selection, which is a caving rock, it's showing at 0.3, which is also moderate heritability. That when you get this number, before you make the selection, you had hope that you can get some gain from this selection. And of course, I make some gain from this selection. These graph represent, the y-axis represent the percent germination, and each pair of the bar graph represent each cycle of selection. On your left-hand side is the response from the caving rock, and your right-hand side is a shelter. Let's see at the caving rock first. That you remember that I got moderate heritability, so I should have some progress here. The yellow bar is the control seed, and the blue bar is the inoculated seeds. As you can see here, for the base cycle, the cycle 0 that never be selected before, this is significantly different between control and the inoculated one, that the inoculated one has a reduction in the germination. And the difference between the control and the inoculated one still persists in cycle 1 also. But these differences between the control and the treatment one was reducing into non-significantly different within cycle 2, which is good that, okay, you get the good result out of this. The similar trend is also happening with the shelter, that the base population still have the significantly different between the control and the treatment one, but within just one cycle of selection, that gap was significantly different at all here. And based on this gap between the control and the inoculated one, I plot this gap as a % germination reduction. The lower the bar, the more resistant this is. As you see here, the trend is very obvious that from cycle 0 to cycle 2 in Kevin Rock, it's dropped like this, which means that it's increased the resistance, obviously. The similar trend also happened in shelter too, that it's dropped, but it has a variation here, but it's already like different than the base population in this case. Based on this huge improvement on % germination reduction, the % genetic gain between cycle 0 and cycle 2, here, it's over 100%, but it's around 60% per cycle of selection. And it's also around 50% per cycle of selection in shelter also. And based on these % genetic gains, I can calculate the liability heritability, which is the realized heritability in this case of the binary trait like this, that it's got 0.2 here and 0.3 here, which is acceptable moderate heritability. So as a plant breeder, I'm so proud of this result that, okay, I got something out of this. But what about, do you remember another disease, the biphoid leaf spot? That is like, so I got this result, so I hope that I get something from that. So I went to the similar step to first evaluate the heritability of that resistant to biphoid leaf spot. And this is the result. This is like, am I dreaming? Is it 0 non-heritability? Are you serious? Like I spent like 3 years on this and it's 0. So, but I didn't give up that time. But I gave up already. Just kidding. So at that time, the first evaluation of the estimate of the heritability estimate was done in 7 trays with 3 replicates. So I tried to like change the inoculation method from just the hand spray to the airbrush that can produce a more consistent small drop of the inoculum that can firmly attach to the leaf well. I tried everything. And I also try to fit just from 125 hapsid progeny across 7 trays. I fit it into just 1 tray and just discard. Like most of them just use 44 hapsid of that. And I try like lots of combinations of here. And I spent almost a year on this. And wow, all of them are 0 or non-significant. That's just great. That's just very good news for you. So and this is like, but I didn't trust the result from this heritability estimate because, come on, that's just the estimate. And what about if I just stubborn to make selection based on these techniques? And this is the result of my stubbornness. that none of them, none of them looks the same. Are they copying the result to each other? Like none of them making any progress out of this selection. So what's wrong with that? I still don't know. So I try to make the test cross, like to go back to just your basic genetics class, to go back to your first step, like to make the test cross. I make the cross of the resistant by resistant, resistant by susceptible, susceptible by resistant, and resistant by resistance from the basic textbook. And I found that all of them, like these are the histogram of the progeny of each cross, the resistant, susceptible, and each cross of here. The top here is a cave rock progeny, and the lower here is a shelter progenies. And all of these graphs like showing like almost normally distributed with just unique model pattern. Like there's no multiple like peaks in this segregation in this histogram. So I can conclude that these resistance should be controlled by multiple alleles with the small effects of each allele. And because there's no significant difference between the reciprocal cross, I can conclude that there's no linked inheritance here. And also, there's only like, even like when I try to compare between these groups of the cross, only the resistant by resistant is different from the susceptible by susceptible, which is not, this difference is expected. But I also expect that the one that got the resistant in the cross, like resistant by susceptible and resistant by susceptible by resistant, should be different from the susceptible by susceptible also, but it didn't show that way. So in the conclusion from this selection, based on the different trend in the selection, that the top bar is a selection, it's a screening for the biparate seed rot, and the lower result is a resistant to biparate leaf spot. And you see like there's no trend here, like flat trend here. And in the resistant to biparate seed rot, there's a huge trend here, like it reduced here. The light blue bar here representing the population that was selected for biparate leaf spot. So although there's a reduction here, there's no significantly different between these with the base population. So at this point, I made significant progress in the resistance to biparate seed rot, but I didn't make any progress in the biparate leaf spot. But that's not all the bad news, because do you remember the previously that I mentioned that this disease was inoculated firstly in the seeds. That's the seeds carry the pathogen into the field at the first. So this at least a good news that I can reduce the primary inoculation to the field at the beginning. That is overall going to reduce the incidence of biparate leaf spot in the field in the later year. And what's the explanation of the no progress in resistant to biparate leaf spot? There's a bunch of them. There might be the low natural pressure of this, or this is a complex trait that it's controlled. Like this is, and I work with the octoploid switchgrass, and 2 cycles of selection may not be enough to push these to be more resistant. Or the worst case is there might not be the resistance in this population that I'm working with. So here is a conclusion for this part of my research, and this still confirms that there's no correlation between these two resistance, and this can give an idea that in future we can make the selection based on these 2 traits like separately. So, but at this point of my result, I was at my 4th year already. So if you add in my shoes, so you know how it feels. But I didn't feel discouraged. Actually, I feel a bit. But I try to learn what's wrong with the resistant to biparate leaf spot. And because even though this biparate leaf spot, it's one of the major disease in rice, but there are only 4 publications about the resistance in the rice population, which is quite, I don't know what's wrong with this, but there's only 4 papers of that. But the most recent paper was trying to identify the resistance in 3 chromosomes, and they use SNPs. They can confirm the 13 SNPs marker that linked to the resistant region here. But they just, in that paper, they just stopped there. So I tried to dig a little bit deeper. That in that paper, in that research, they used the near isogenic line between the indica and the temperate japonica. And based on that, like I just want to know like what are the resistant genes that link to these markers. So the linkage disequilibrium in indica is around 100 kb, and the LD for the temperature ponica is 200 kb. So I just use the 200 kb 200 kb linkage disequilibrium here to just find what are the genes that link to this 13 SNPs marker. So I just searched it in the phytosome database, and I found that there are 278 genes that linked to these 13 markers, and half of them are annotated, another half is not. And interestingly, most of the frequent hit that was showing to me, including the leucine-rich repeat family protein and the nucleotide binding leucine-rich repeat here, also like showing the most out of 278 one. And these two proteins are well known for its function as a R gene because it's detecting the pathogen. Another most hit protein is the peroxidase precursor. Peroxidase is quite important in terms of the resistance for these pathogen to this pathogen because the Bipolaris oryzae is a necrotroph, so it used the programmed cell death that related to reactive oxygen species in the mechanism of the susceptibility. So this is important for that. Diligent is also important in terms of the lignin synthesis that it prevent the infection for this disease. The MYB protein is very well known for its resistant capacity that it responds to most biotic and abiotic stress in general disease. So based on these 287 genes, I just pulled up all of the sequence and blast back to, so because this is based on the RICE data, so I pull out of these gene sequence and blast back to the Switchgrass database, and I found that the top 10% score of those blast hits, there's still like 1,400 genes out of that. So at least I have some hope that there should be some resistant genes in Switchgrass population. So at this point, I try to dissect the resistance to Bipolaris leaf spot by using the Genomewide association. So I went back to the first step that I would not working with the seedlings anymore. It gave me everything. So I worked with the mature plant that I've got a chance to evaluate these in the mature plant that was already a genotype. So the population that I use is a Switchgrass Northern Association panel that consists of 66 population that both the commercialized cultivars from Switchgrass or some of them are also the natural collection of the Switchgrass. And from those population, we plant 5 to 6 seeds for each population into individual. So in total, I have 479 genotypes out of this. And in 2016, we planted in Ithaca and in Phillipsburg in Pennsylvania for 3 clones. And just last year, we was just planted in Rucker. So, but these data, it's only from last year data because I only have 1 year left. So I was having only one chance in 2 locations. So at this point, with the 66 population 479, I just want to, the goal was to screening to just identify what's the best candidate for the resistant out of this population. And what are, because all of these has been genotyped already, what are the marker, the SNPs marker that links to potential resistant gene to the Bipolaris Bipolaris leaf spot. So at first, it's the easiest way to evaluate the disease. Just walk through the field, right? So I evaluate by just using my eyes, by evaluate with the score from 0 to 5 in the field. And the result showing like this, that so I represent these results in 66 population. Each bar represents 66 population. And it's coded by the different ecotype, the Upland and Lowland. And these bar order based on the mean from 2 locations. And as you can see here, there's no similar trend between the based on the mean from two locations with the mean from just New York and mean from Pennsylvania. And that's showing me that there's a huge environmental effects to the resistance to bipyrite leaf spot. So I come up with so I try to evaluate, I try to eliminate those environmental effects by just taking the leaf sample back to the lab and sprayed it and inoculated it. So I did into two assays, in the leaf detachment assay and the leaf dis assay. For the leaf detachment assay, I basically just cut to 5 centimeters, surface sterilize, sprayed it cover 7 days and evaluate it, and also like scan each leaf, scan each leaf and use image analysis to detect the lesion. So you may notice that like I use the disease is in the dot, right? The black dot. But so I can, based on these pictures, I can extract lots of data out of that, just the black dot or just the overall necrosis. But in this one, I show you the overall necrosis. That showing, and that result is pretty high correlated with my eyes evaluation. But I know that's not a good like candidate from the result that I give you, like non-significant result previously. Another evaluation that I did was using the leaf disc assay, that I just core the leaf into 1 centimeters and put in the water agar and take pictures of that and use image J to extract each leaf circle and evaluate like the similar things also. And that's a high correlation at 0.7 with like correlate with my eyes result. And based on these, I can evaluate. So I evaluated broad center heritability, and this time it's showing very high broad center heritability at 0.7, 0.5. Skip this one, and 0.3 and 0.6. I skipped this one because you remember that there's a huge environmental effect here, that you have the high heritability in each location, but when you consider two locations, where are you? So that's the point here. So even though I have this high heritability from each evaluation approaches, but when I try to just rank it by the DTIIA, is the result from the detached leaf with the image analysis one. So I rank this based on the detached leaf results, and you see that there's still like no trend with this leaf, and no trend with the mean of two locations. And this showing that like there's no correlation even with within the evaluation for the resistant to bipyrite leaf spot. There's no correlation between that. Like this high correlation is between my eyes and the vision. And beyond the resistant to bipyrite leaf spot, there's also like no correlation with other biomass traits, such as the mineral content, the digestibility, the cell wall concentration, or the ethanol yield from the dry biomass. So based on the fact that there's no correlation, based on this, the way that I fit the GWAS, I was trying to just fit it as by itself, by its single trait, or just a combination of the lots of traits. And I also include the first three principal component that can explain around 15% variation in this population, and also use a kinship matrix as a random effect too. And the LD, the linkage disequilibrium, that give the idea like how wide is the gene that linked to the marker that I use. And in this population is 20 kilobase pair. So as I mentioned earlier, that the way that I fit the GWAS is I was trying to just fit just a single trait of itself and the combination of many traits. So from the detached leaf, from this leaf, from the field and everything. And more than 90% of my GWAS give me non-significant peaks, and like or even though, or some of them just like give me the very unreliable QQ plot, that some of them left the panel too early, some of them just like smoothly normal, didn't show me significant. But some of them still show me like some hope, that's still something in my research. So I also, based on, because I have like 479 population, I subset in those 479 genotypes into like many groups. The first one is just I use like the whole thing with the combination of the detached leaf and the disleaf and the maximum in New York State. I use maximum the value from the field evaluation, which is the most severe in the field, because none of the blob or none of the mean from the field evaluation show me the significant results of the GWAS at all. And I use the maximum because Washington in plant pathology suggested me to do this, that, hey, you don't need to think as a plant breeder all the time. You need to think as a plant pathology sometimes, that if you have the plants in the same field, that you have 3 replicates, one of them show no symptom, one of them show the worst. Why don't you just pick the worst one? Like consider that's the worst case. So that's why I consider using the maximum value here in this case. And it's showing something here, that some significant peak here is related to antioxidant protein, which is very good in terms of the resistance to bipyrate leaf spot. MYB, which is related to the biotic and abiotic stress. This one, the ring finger, this is related to program cell death. And GST, the glutathione S transferase, is also related to the antioxidant activities. The most important peak here is this one. I know that it's not like my GWAS didn't provide like the very Manhattan plot, like the significant peak like that, but this showing me the most interesting one, because the cytochrome P450 is the only one peak that overlap with the blast result that I got from rice, that I used the gene from rice and I blast back to the switchgrass. And this is the only one that overlap with that data. And the importance of this phytochrome P450 it's synthesized for triterpene. Triterpene is also well known for like the antifungal activities. The most well known triterpene is abedicin in oat, that is also like also resistant to act as a antifungal activities too. Another subset is the tetaploid in this switchgrass. So I fit this. So the tetaploid showing the only GWAS that's showing the significant from the tetaploid subset is the maximum value in the field in New York State, and it's showing that it links with the programmed cell death protein and also the protein that induced the necrosis one here. Another subset is the subset of the lowland, that it also shows like the antifungal proteins here, and here it's linked with this peak linked with the nucleotide binding site leucine rich repeat protein, which is also well known for acting as an R protein that detect the pathogen. So for the upland, it's showing only one peak here that linked to DW40, even though there's no like study like directly say that this is like related to any plant disease, but it's also, but this is very important for biosynthesis for MYB protein, which is also linked to the plant stress. And based on this, although like as you know that like I have most of my GWAS non-significantly different, like didn't give me significant result. But the significant GWAS that I have here, there's no overlap peaks out of this population at all, which is it's not an ideal result, but at least it's better than just zero heritability and non-progress from the selection. And they're showing up like the key that they're showing up that these, and when I compare, consider overall of these GWAS, there's they're showing up the key genes that link to these markers that is reasonably that can confer the resistance to to this disease. But it's still missing some genes that I expect to see, like diligent that is linked to the lignin synthesis, the peroxidase, MLO like proteins, and the only peaks on chromosome B from the 479 here, it's linked with the blast result from previous from the rice. And based on this, let's get back to the goal for the study of this. That the first goal is trying to identify the most resistant genotypes or population in this 66 population. But as you remember, that there's like none of the phenotypic approaches here has correlation between each other. That's why like I cannot make the conclusive decision that okay, this one is the best out of all of them in all criteria. But if I need to pick the best one right now, I will pick SW805 because it's always on the top 10% of this population. And the multi-thread GWAS here really helps me to review some of the SNPs marker that is linked to the resistant gene reasonably. So at this point, it's the end of my breeding part that I try so hard to breed for these traits. But don't forget that we still can manage this disease by another approaches. And in this recent year, there's a trend to implement silicon to rice to be resistant to Bipolaris leaf spot. And it has been proved that this silicon can coat the seeds. Can coat the leaves to prevent the attack of the intrusion of this pathogen like physically. And also this silicon can trigger the defense mechanism molecules such as the jasmonic acid or salicylic acid. Lastly, it's quite surprising that they also prove that the silicon can prevent the taking over of the ethylene production from rice. So based on these, there's a study to screen the gene in Canlo switchgrass, and they found that there are at least two key genes in translocating for silicon in switchgrass, LSI1 and LSI2. So based on this knowledge, I had some hope that okay, even though I didn't make any progress from the selection, I can make progress from just applying silicon to switchgrass and make it more resistant here. But there's a concern about applying silicon to switchgrass too, because switchgrass was aimed to use for the biomass, right? So we basically want to burn it or turn it into ethanol. If we want to burn it, the higher silicon amount can melt at 500 or 700 degrees Celsius and clog this and create the slag like this that can clog the heating system. So that's like the implement of silicon into switchgrass is also a concern too. And these are just the basic idea of how much silicon in the tissue of this plant. And you can see here like switchgrass is already higher than miscanthus, which is also another biomass crop too, but it's still lower than rice. So maybe just add a little bit more silicon may not hurt this, but which is not a good sign. The result showing that there's no effect of the silicon amendment to the resistance to either diseases. I try to mix the silicon powder with the soil and just plant switchgrass there and taste the Bipolaris seed rod there, taste the Bipolaris leaf spot there. There's no significant effect on that, even though like the yellow bar here represent the silicon one and the blue bar is the control one. Even though it looks like significantly different, but when we taste statistically, there's not like significant difference. And so, okay, there may be like some soil mixing problem. So I try to just water the silicon solution to them too, and there's no improvement between silicon and the control one. So at this point, I think I'm cursed to this disease somehow. Just kidding. So there's no improvement, but there's quite some explanation on this too. That there might be a low retention of this silicon in the. I didn't use the soil. I used a corneal peat-like medium, so there may be low retention on that. And like there's no biological, like for the resistant to Bipolaris seed rods, there's like watering silicon when it seeds, they may not have any biological effects on that. Lastly, like the stage that I use for screening for Bipolaris leaf spot may not be like enough. They may not be appropriate for the silicon to accumulate like in the tissue already. But we need to keep in mind that these results, it's a greenhouse result, so it cannot be inferred the effect of this silicon amendment in the field. So if anyone want to try this, like you're welcome to continue this research. So overall, for my research, I would like to conclude that as a plant breeder, I'm pretty proud of the improvement of the resistance to Bipolaris seed rot. That it's increased like, come on, 60% and 50% of each cycle of selection is so high for the disease resistant. But I have to admit that the resistance to Bipolaris leaf spots still need to be improved, and since the screening techniques, and even though I try to screen in mature plants, it's still like those markers that come from the multi-trait GWAS cannot directly be used for the genomic assisted selection right now. It still need to be validating a bit. And lastly, for silicon amendment, we cannot infer the greenhouse result to the field result. So that might be the future research again. Lastly, I cannot stand here and live in America for 5 years without the financial funding from the development and promotion of science and technology from Thailand. And also, I cannot be here and make sense of my research without my committee member, Don Van, Gary Bergstorm, and Neil Matson. Also, I cannot plant all of those 500 of those plants in the field every year without the help of the forest group, Julie Hansen and Jamie Crawford, Brian Crawford. And thanks to the member group in Gary's lab for helping me with the disease part, and also the plant material and the genotype data from Mike Kessler, and all of my SIP friends, that I cannot have all of this idea by just sitting by myself. I got lots of ideas from you all. So thank you so much for having me here. And welcome for any questions. And if you want to have any comments, very welcome to email me if I too. Thank you so much.
If there's any quick questions, or somebody has one that's going to volunteer, you can come up and talk with us. Anybody have just one quick question you can ask?
How expensive is the silicon amendment? It seems like that might be an additional objection.
How expensive is silicon? Neil, I didn't follow the actual price.
I've done work in container media, and it would be like a 0.1 cent per 6-inch pot. But I'm not a field person, so I'd have to think through how to scale up for field. I do know there's different ways of adding it, and one way is actually using rice hulls or etched rice hulls. So I think the ideal is to use these, like where our silicon already occurring, that's in waste streams that we can put back into agricultural production. Thank you.
Again, if there are any additional questions, feel free to come up and talk with Chris. Other than that, just well done.
Thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.